Good morning. Thank you for joining us for our second Cell Health webinar. My name is Nick and I'm in the R&D team here at Molecular Probes, part of Life Technologies. I'm joined this morning by Ed Lieber from our technical support team, so feel free to ask any questions as we go. We'll do our best to answer them live, but if we can't answer them as we go, we will get back to you via email. Okay, so the topics we're going to cover today, we're going to touch briefly upon some of the properties of apoptosis, essentially what happens and when. We'll also look at some of the parameters involved in apoptosis, so essentially what is it possible to measure. And then we'll delve into a little bit deeper into some of the probes that you can be used to investigate apoptosis. These are basically some of the fluorescent molecules that exist that will report these changes that we've discussed earlier. And then very briefly at the end we'll try and cover a few of the tips and tricks to optimize your experiments. So as we go into talking about properties of apoptosis, in our first webinar on cell health, we talked about probes that really just detected whether or not the cell was live or deaf, and then introduced this concept of vitality and viability as a continuum between life and living cells and dead cells. Today we're going to talk about apoptosis, and as I'm sure many of you know, that apoptosis is really on the right-hand part of the continuum. It's really the program mechanism to bring about cell death. And so we're going to look at how this occurs and how it can be measured and assayed, primarily using fluorescence detection. So apoptosis really just means off and away and falling. So it was originally used to describe the falling of petals from flowers or leaves from plants or trees, but it was adapted into a biological context, and that's really its primary use of the word nowadays. It's to describe the programmed death of cells within an organism. So some of the features of apoptosis. It's biochemically and morphologically distinct from necrosis. This is a very simplified view. There are areas where they overlap significantly, and so it's important when you're choosing your assays, if you're just wanting to measure necrosis or apoptosis, or indeed maybe both, it's important to make sure that you're picking characteristics and criteria that are distinct from one another. To talk about apoptosis a little bit here, some of the biochemical features are the fragmentation of the genome and cleavage of cellular proteins, and we'll go into a little bit more detail of that later. There are also key morphological features that can be assayed. These are compaction of nuclear chromatin, formation of membrane-bound apoptotic bodies, shrinkage of the cytoplasm, as well as loss of membrane asymmetry. Mechanistically, apoptosis is a fairly well-defined pathway. There's extrinsic pathways, intrinsic pathways that bring about apoptosis, as well as the granzyme B pathway. Essentially what happens with the extrinsic pathway is there are death receptors, for example, the FAT-L TNF-alpha. TNF-alpha stands for tumor necrosis factor alpha. These are death receptors. They activate FAD, which goes on to activate a cascade of intracellular caspases, here denoted by C for caspase, and then a number for the caspase 1 through 10. The extrinsic pathway goes on to act on BID and then truncated BID, TBID. This promotes Back's association with mitochondria, which causes loss of cytochrome C from mitochondria. This pathway can also be activated via BH3 domain proteins. This is the intrinsic pathway. The BH3 containing domain proteins are normally inhibited by members of the BCL2 family, and indeed removal of that inhibition will activate the intrinsic pathway. But as I say, they both lead to Back's recruitment of mitochondria, loss of cytochrome C, as well as loss of mitochondrial membrane potential. The loss of cytochrome C basically nucleates the apoptosome. This is a combination of caspase 9 and A path. They're in a 1-to-1 stoichiometry. And the apoptosome activates caspase 3 and 7. And indeed, caspase 3-7 assays are one of the best ways of looking at apoptosis. It's a real hallmark of the induction of apoptosis. And these caspases go on to act on further cellular caspases. We'll talk a little bit about what they do in a moment. So to touch very briefly on the parameters and the properties. Well, the cellular functional changes that we see, one of them is the phosphatidylserine translocation. And this is what underlies the loss of membrane asymmetry. The functional change can also be the activation of caspases, we talked about a little bit earlier. This is the activation of these proteolytic enzymes, and they attack key intracellular components, things like the cytoskeleton, as well as the nucleus, and they can also cleave other intracellular organelles such as the ER and Golgi, and they essentially bring around the compromisation of normal cellular function. Plasma membrane integrity will also be lost, and this leads to breakdown of main cellular structure and causes the leakage of intracellular components out of the cell. It's important to remember that plasma membrane integrity, or a loss thereof, underlies other forms of cell death such as necrosis, and this is a key indicator that we use across a lot of different cell viability assays. Then the second functional change that we see is DNA condensation and fragmentation, and so this is really just DNA strand breaks. This is a relatively simple process to assay and to start to look at. This is at a very high level. It's important to remember that there are some far more complex traits that exist, and they can be a little more cell type specific. We're trying to deal in very general terms today, look at properties that are conserved across all cell types. But it is important to remember you may see cell type specific ones, and indeed, even within general properties, there are some more complex ones that we won't touch on so much today, but we're happy to answer questions if anyone has any very specific questions about a very certain aspect of apoptosis. And this table here also represents a rough representation of the chronology of apoptosis. So it's important to remember that these things don't happen all at once, they occur in a progressive fashion. This is from the model of camptothecin-induced apoptosis in Jurkats. It is only rough, and it is for Jurkats, but it is a good example of the flow and how it's sometimes important to measure early and late stages of apoptosis. So at the top we have some of the early events, this loss of the membrane asymmetry, and then followed by Caspase activation. There's also differential permeability of the plasma membrane, which is distinct from actual complete loss of the membrane integrity, and we'll touch on an assay using one of our dyes, YoPro, later. We also can see nuclear condensation following that differential permeability. We then see the formation of a sub-G1, G0 peak in DNA, and then followed by DNA fragmentation and membrane integrity loss. And some of these things are happening pretty similar time courses, so nuclear condensation and the sub-G0 peak formation are fairly closely tied temporally. So again, just if we delve back into that table, essentially what we can do, we can start to look at the parameters and some of the assays that will feed off that. So loss of membrane asymmetry, so the assay is looking at membrane orientation, so there's various different probes that will pick up subtle changes in the membrane composition and orientation. The Caspase activation, you can assay the activation of proteolytic enzymes. We have a number of different assays that work either in cell systems or cell-free systems. Compacted nuclear chromatin can easily be picked up with various nucleic acid stains. The shrinkage of the cytoplasm can be picked up with general cellular stains, and indeed if you want to look at fragmentation of other organelles, there's very many good antibodies targeting fluorescent proteins, or indeed organic dyes that track various cellular structures like Mitotracker and ER tracker and some of the labeled ceramides for the Golgi. Production of apoptotic bodies, similar assay to what you would do for compacted nuclear chromatin. You can use nucleic acid stains. And then finally, again, with chromosomal fragmentation, you can pick this up with assays that look at DNA strand breaks. So that's a little bit of the introduction sort of over and done with, and we're going to dive into the nitty-gritty of the probes that one would use to look at apoptosis. We'll focus on those assays that are primarily involved in fluorescence detection. This is those that will work on wide-field or confocal microscopes, flow cytometers, plate readers, or indeed high-content imaging platforms. We'll look again at the primary assays that report the hallmarks of apoptosis, ones that are applicable across a broad range of cell types. We'll also delve a little bit into how we can multiplex with pre-lethal indicators as well as stains for total viability. We'll have a separate section at the end where we look at a couple of pre-lethal indicators, and you'll see throughout the presentation we'll be using multiplex data with viability stains. So one of the first assays and the first points of apoptosis is this loss of membrane asymmetry. Normally in the cell, phosphatidylyl serine is on the cytoplasmic face of the lipid bilayer that is the plasma membrane. In an apoptotic cell, this lipid bilayer will flip, externalizing phosphatidylyl serine. Now, annexins will bind to phosphatidylyl serine, and if you label those annexin with a fluorescent conjugate, you can detect them using flow cytometry. It's important to note that a late apoptotic cell or a necrotic cell will have a compromised plasma membrane, and so fluorescent conjugates of annexin can enter the cell and bind to phosphatidyl serine that's on the cytoplasmic face and giving you false positives. That's why it's important to combine these probes with indicators of cell viability. Here we've used a Cytox dye. This is a cell-impermeant nucleic acid dye. This allows you to discriminate between truly apoptotic cells versus late apoptotic or necrotic cells. It's a very important control to have when doing these experiments. So what does this data actually look like? Well, here we've got Jurkat cells. So these are immortalized T-cell line. They've been treated for 4 hours with 10 micromolar camptothecin. On the left we have our control cells, and as you can see there's a very dense grouping of cells that are negative for annexin fluorescence. Here we've used one of our specific blue conjugates, but we also have the full range of Alexafluor dyes conjugated to annexins. And they're also negative for Cytox AAD advance. This is a cell-impermeant nucleic acid stain. These are viable. At the top here we have, in the red top right, is the necrotic population. Those have compromised cell membranes, and so that's why you're seeing some annexin fluorescence as well, because it's binding to non-externalized phosphatidyl serine. However, if we look at the chart on the right, after induction of apoptosis with camptothecin, we start to see a significant increase in the cell population that is apoptotic. And again here, just to reiterate this, we're defining apoptosis here as being able, annexins binding to the phosphatidyl serine, so these have been externalized. However, they have no loss of membrane integrity, as there is no increase in the staining with a cell-impermeant nucleic acid dye. Here we're using, as I say, Cytox AAD advanced. There is also a little bit of an increase, or at least a shift in the necrotic population as well, and it's important to note that even drugs that induce apoptosis can also just lead straight to a necrotic death, or just straight into very fast apoptosis, which will give you that late apoptotic necrotic phenotype. Another way of gauging this loss of membrane asymmetry is with the very eloquently named F2N12S dye. This works on the same principle of externalization of various phospholipids, phosphatidyl serine and phosphatidyl ethanolamine. These translocate during apoptosis to the outer leaflet of the plasma membrane, and this causes a change in surface charge. And what F2N12S senses is this change in surface charge. It's a ratiometric probe, and it's self-calibrating, so it's independent of probe concentration, which is very nice if you're trying to optimize an assay relatively quickly. Again, if we have a look at what some of the data actually looks like, again, we've got Jurkat cells that have been treated with camptothecin for 4 hours, and I believe it's 10 micromolar concentration. And here we've got 200 nanomole of the probe F2N12S. In the top of the graph on the left, you see the orange fluorescence against the green fluorescence. As I say, this is a ratiometric probe. It has two emission bands, one green, 530, and one orange at 582. It's a violet excited probe for those flow cytometry aficionados in the audience. You can use a ratio of the orange over green fluorescence, and in light you can pick up live and dead cells there. If we then treat with camptothecin, down on the bottom panel there, what you see is that change. You see a loss of the orange fluorescence and an increase in the green. It's read out as a change in the ratio, and you start to see that apoptotic population appearing in the bottom quadrant. And again, we've used a cell-impermeant nucleic acid dye to discriminate between early and late apoptosis. So moving on in the chronology of apoptosis, from loss of membrane asymmetry to activation of caspases. We have a new reagent, CellEvent. This is a fluorogenic sensor for caspase activation. Essentially, when caspases are activated, the probe is cleaved and becomes fluorescent. Just to show you here, we've got a dose response of staurosporin in HeLa cells. And so we've dosed in increasing concentrations of staurosporin, and you can see an increase in the fluorescence from this signal. Here we haven't actually done mean fluorescence, we've gated on positive cells, so you're seeing an increase in the number of cells that are positive for caspase activation. But this also works as a purely fluorogenic sensor. The data here is from a high-content imaging platform. It does work for flow, which I'll show in a moment, as well as plate readers and traditional imaging. And indeed, on the right here, we've got a population which is treated with low-dose staurosporin for 4 hours, about 0.5 micromolar. And we can see one cell just here on the sort of right in the middle, showing up very brightly for green, which is the caspase activation. The cells are also co-stained with alexafluorphalloidin 647. Actually, that's said the wrong way. It should be phalloidin alexa 647, as well as Mitotracker orange and the CellEvent probe. Again, just a little bit on the pharmacology of this probe. We can show some specificity here for caspases. Using a constant dose of sterosporin to induce apoptosis, we can then dose in an inhibitor of caspases. Here we're using the ZDVD FMK inhibitor. And as you can see, as you put in more and more of this inhibitor, you lose the signal from the caspase probe. Again, showing its specificity that it's actually picking up caspases. On the left we have the no inhibitor, and on the right we have our 20 micromolar inhibitor, showing the complete loss of the signal. And as I said, it works in flow cytometry. So the nice advantage of this probe, it's fluorogenic. It contains the specific caspase 37 motif DEVD, and so it is truly specific for the activation of caspases. And on the left we have some control cells. Again, these are Jurkats, one of our sort of workhorse cell lines for flow cytometry. And on the right we've treated them with camptothesis, and you can see that increase in the apoptotic population with no increase in loss of membrane integrity, which would belie a late apoptotic cell or indeed necrotic. So we can show that these are truly apoptotic and early apoptotic cells at that. And again, these just further highlight the need to include a dead cell dye in these experiments to get that multiparametric readout of apoptotic, late apoptotic and necrotic. There are other ways of monitoring caspase activation. Here we have our FLICA technology. These FLICA reagents are fluorescent inhibitors of caspase activity. Essentially what happens is you load the cells with the FLICA reagents, they get into the cells, and if there are any caspases that are active, these inhibitors can bind to them. They'll only bind to active caspases, and once bound they can't be washed out. And so if a cell has active caspases, they'll retain the fluorescent signal, and if the cells don't have active caspases, the signal will be lost. These are part of some of our Vibrant FAM caspase kits, and these are particularly useful for flow cytometry. They do work in imaging, but they're primarily used for flow cytometry. And again, we can pick up this FAM positive but propydium iodide negative population, which is a truly apoptotic, early apoptotic population. Also in cell-free systems, if you take a cell lysate, you can use some of our Quenched caspase substrates that pick up different motifs. So these peptide sequences listed on the left are specific for various caspases. So you can see here the DVD, which is mainly used for caspase 3-7, but will also be cleaved by caspase 6, 8, and 10. Some of the other sequences there, VEID for caspase 6 as well as IETD. And then some of the generic aspartic acid residues that will be cleaved by a lot of different caspases. And then some peptide sequences for caspases 1, 4, and 2 up at the top. And almost all of them are using green fluorophores, the rhodamine 110, although we do have a blue AMCA fluorophore there as well. And these are, as I say, only work in cell lysates, but they do give you a lot of flexibility to assay specific caspases. So as I mentioned earlier, there are two different aspects of membrane permeability and apoptosis. There's this complete loss of membrane integrity that is the hallmark of both late apoptosis as well as necrotic cell death. But there's also a much less well understood differential membrane permeability, and it seems to be specific to certain dyes. And one of them is the dye Yopro. Essentially, on the right this time we have untreated cells. You can see in the yellow scatters are apoptosis. There's very few cells that are apoptotic here versus the green viable or just completely dead in red. However, if we treat these cells with camptothecin, we see an increase in Yopro fluorescence. It's an increasing number of cells in that yellow circle, but there's no increase in the PI staining. So this is not a complete loss of membrane potential, which lets the normally impermeant Yopro in. It's a differential plot. It's a differential permeability. So, and as I say, the mechanism is not well understood, but it's fairly well used in the literature as a way of teasing out just that subtle change in the membrane permeability. And as I say, I see quite a few questions coming up. Hopefully we're getting to them as we go through. But as I say, if we don't get back to you straight away, we will get back to you not long after the presentation is finished. Looking now at assays for nuclear chromatin condensation. Again, nucleic acid dyes give a very nice readout of these. Here we have some camptothecin-induced Jurkat cells, and we can see that increase in the dye cycle fluorescence that is showing that the nuclear chromatin is condensing and giving a much brighter signal from some of the nucleic acid dyes. And if you look, for example, on our website, we can see a full range of nucleic acid dyes that span the visible spectrum. I'm just going to address one question here. We've just seen one that comes in showing that we don't show the events that are positive for the cytox but negative for the apoptotic markers. Those events would be truly necrotic because you wouldn't have any of the apoptotic loss of membrane integrity. You just see sort of bulk loss of membrane integrity. And these are some of the assays that we covered in the previous webinar on tools for viability. And later on, at the very end, we'll do some tips and tricks which suggest some dead cell control. So it's actually ways of killing cells very rapidly without inducing apoptosis. But anyway, back to detection of sub-G1, sub-G0 population of cells. So this is a little bit of a busy slide. I apologize. We toed and froed about what to put on this slide and what to make it a little bit overwhelming versus getting a lot of information in. Hopefully we strike the right balance. Anyway, on the left here, up top left, we've got what you'd see for a regular dye cycle profile. This is DNA. And down here we've done some mode fit analysis. What you see, that tall peak on the left, that's your G1. That clear and then hatched line in the middle, that's the S phase cells, and on the right is the is the G2M peak. So you can tell by the intensity of DNA dye staining what phase of the cell cycle the cell is in. If the DNA starts to fragment and is taken into apoptotic bodies, you get less DNA than you'd get in a normal organism, and so the fluorescence is much, much lower. So that's what you're seeing on the right here. We've got camptothecin cells, and we've treated these ones for a little bit longer, 6 hours, to get complete nice signal of this fragmentation and encapsulation into apoptotic bodies. But you can see this peak appearing, the blue peak to the left. This is DNA staining that is less than what you'd see in a normal 2N cell. So this is a fragmentation of the DNA and the formation of a sub-G1 population. Other assays for chromosomal fragmentation can be done by using the principle of DUTP incorporation at the 3' end of damaged DNA. The enzyme terminal deoxynucleotidal transferase will add in UTP residues at the 3' end if DNA is damaged. Now we can take advantage of that by using fluorescent analogues of UTP, things like EDUTP or BRDUTP. And they will be incorporated into damaged DNA and then can be detected through fluorescence. And we have an example of that on the next slide. Seeing a question about flow cytometry with attaching cells. So adherent cells you can do flow cytometry on. However, you do have to be very careful that when you trypsinize them or scrape them, that you're not sort of damaging the membrane integrity or indeed causing cell death itself. But it is possible. You just have to be very, very careful, and it will take a little bit of time to optimize. In terms of chromosomal fragmentation, as I said, we have some assays. The assay I'm going to talk about mainly here is the click assay. And actually, I'm just going to go back to the previous slide. As I say, damaged DNA will have DUTP incorporated. We can have a clickable analog. This is EDUTP. It's incorporated into damaged DNA. And then once cells have been fixed and permeabilized, you can come in with an ElexaFluor detection reagent. The EDUTP is an alkyne. You can have an ElexaFluor azide, and then there's a copper catalyzed reaction between these. This is the click reaction, which forms a very stable covalent linkage. It attaches a fluorescent moiety to anywhere that DNA damage has occurred. And if we just go to the next slide, actually see an image of this. So in orange we have microtubules, and then in blue we have an antibody against cleaved caspase, in green, and then Herxt in blue, and then tunnel is the magenta signal that you can start to see in that very top cell. And so you can start to look for chromosomal fragmentation. And indeed, you can do this in other platforms. Here's a high-content image, untreated and treated. So we've treated these HeLa cells with 2 micromolar staurosporin for 4 hours, and you can see the appearance of this magenta signal from the Alexa Fluor 647, which we've flipped to the EDUTP in the tunnel reaction. It's a very nice reaction for looking at chromosomal fragmentation. And the primary advantage of a EDUTP over BRDUTP is speed. The click reaction is complete within 20 to 30 minutes versus a BRDU assay, which can take several hours to 24 hours at an extreme. Okay, so some of the multi-parametric approaches that can be taken. As I said, the previous slides have done a lot of multi-parametric analysis with dead cell dyes and apoptotic probes, but here we're going to look at a few subtle dyes that will look at some of the pre-lethal indicators. Here we can look at the induction of apoptosis at the same time as looking at generation of reactive oxygen species. Here we're using a dye called CellRox Deep Red. It's a far red indicator of reactive oxygen species, as well as the CellEvent probe. And we can see on the left here with staurosporin treated A549 cells, and these were treated with 0.5 micromolar for 4 hours. We can see the purple magenta signal. This is the generation of reaction octave species, as well as a few apoptotic cells appearing in this population. And indeed, that's sort of borne out with the data on the right here. We see a very fast burst of reactive oxygen species generated upon staurosporin treatment, followed later by the induction of caspase. And the induction of apoptosis through the activation of caspases, and this is read out by an appearance of CellEvent positive cells. Here we've got some data looking at autophagy and apoptosis. So we'll actually have a separate webinar that looks at tools to measure autophagy later on in the year. But here's just one example of how you can combine an apoptotic probe with a probe for autophagy. Here we have LC3B RFP delivered with the background gene delivery system. These being expressed in cells that have also been loaded with CellEvent in green and counterstained with Herx to give a feel for total cell number. Induction of staurosporin, you can see, treatment of cells with staurosporin causes an induction of apoptosis and an activation of caspases. And late on in this process, you start to see the appearance in some cells of some autophagosomes as a potentially protective, or maybe as a mechanism of bringing about cell death as well. In one of the early diagrams, I talked about how mitochondria are a key component of governing the apoptotic response. And here we have an assay where we look at mitochondrial health and the induction of apoptosis. Cells have been loaded with 50 nanomolar of the dye TMRN, tetramethylrhodamine methyl ester, and this accumulates in active mitochondria with polarized membrane potential. And we've also loaded them with CellEvent. At the time of adding staurosporin, you can see that we have no green positive cells, so no apoptosis going on, and we have very healthy mitochondria, as all of the cells have very nice red fluorescence. As you monitor these cells over time, you start to see the loss of the membrane potential and the appearance of caspase activation read out by the appearance of green cells. So you can note in the top right-hand corner at about 2.8 hours, you start to see that fluorescence is much lower from the TMRM, and you're starting to see green cells appearing. And then all the way down later at 7 hours, you've essentially completely depolarized all mitochondria, and a large majority of the cells in that field are apoptotic and are showing up as positive for the CellEvent reagent. And we actually have quite a nice movie of this on YouTube if you want to check it out. Okay, so just to finish up very briefly the tips and tricks. It's a little bit difficult to give tips and tricks for apoptosis. Most of these probes require some degree of optimization in your cell type of choice, and that's often just the means by which you induce apoptosis. But we'll try and go over a little bit as much as we can. So for the annexins, it's important to remember that the annexin binding is reversible, and so if you take the annexins out from the solution, you're going to start to lose that signal. You absolutely need divalent cations such as calcium and magnesium for the annexin binding to work. So if you're in divalent-free media or DPBS or HBSS or other buffered salt solution, then you won't get binding. You need to analyze soon after labeling again to get the best signal possible. And you also need to have those controls to make sure that it's truly externalized phosphatidylnyl serine and not loss of plasma membrane integrity. And that's just reiterated. You need to control for those late apoptotic or necrotic cells. And then it's important to note these assays are primarily used for flow cytometry. I've personally done some imaging using confocal with annexins, but they are difficult to tease out a response. Flow cytometry is much better way of looking at annexins, but it is possible with fluorescence microscopy. So because these are primarily used for flow, you've got to remember that trypsinization or mechanical scraping itself can flip that phosphatidylnyl serine and can give you false positives. The next slide just talks about some very general terms for developing apoptotic controls. We tend to use staurosporin, camptothecin, or valinomycin as good ways of inducing apoptosis in a number of cells. Camptothecin works very nicely in Jurkat. We tend to use staurosporin more for cells like U2OS, HeLa, or A549. Valinomycin is also a fairly good drug for the HeLas and A549s, as well as many other different cell types. There are a lot of other drugs. Menadione, for example, is another drug that can induce apoptosis, but a quick literature search using HiWire or PubMed can pretty much find drugs or siRNA treatments that will work for your cell type of interest. The concentration and time course does vary from cell type to cell type, so again, it's important to optimize those yourself and to be aware that just because one study uses one concentration at one time point, it may not work in your protocol. Having those dead cell controls, it's really good to have to show that population. I think this speaks to the earlier question, that population that is positive for dead cells but not positive for apoptotic cells. So heat killing or alcohol killing. Alcohol killing allows cells to be stored or archived for many years. So these are very good controls and very good ways of very rapidly killing the cells. And then just lastly, really, you've got to work hard to preserve the cell structure because a lot of structural changes are brought around by apoptosis and as well as loss of membrane integrity. So trypsinizing or scraping them will disrupt the plasma membrane. You've got to allow the cells to recover prior to trypsinization. And some adherent cells you can actually use non-enzyme treatments. We have an enzyme-free cell dissociation buffer that can be used if you are having issues with those. So finally, we just have a table just to finish off to help you maybe seed some thoughts about which assay to use, determine your equipment, and then look at what step you want to use. So using a flow cytometry, some of the tools that are commonly used is an Annexin kits, F2N1, a 2S, YoPro dye for differential membrane permeability, CellEvent reagents or FLICA and tunnel, all work very nicely in flow cytometry. And they will pick up those events that we list on the right. So Annexins and F2N1 2S will pick up that membrane asymmetry, YoPro for membrane permeability, CellEvent for Caspase, FLICA for Caspase, and tunnel for DNA fragmentation. If you're using an imaging, so confocal, wide field, or even a high content, high throughput imaging platform, CellEvent will pick up Caspase very nicely. FLICA can pick up Caspase activation, and the tunnel works very well for DNA fragmentation. And then high throughput, so these are plate readers. Some of our Quenched substrates that we saw in the table before, these only work in the cell lysates, but they can be done on very high throughput plate readers, as well as the CellEvent reagent, which will work on platforms such as the Flipper. And then finally, a quick plug for our technical support. If you do have any follow-up questions or encounter problems or other areas of concern during experiments, we have online tech support, as well as you can phone the various numbers globally and speak to people in our technical support team who will be able to answer all your questions. So I just finish off by thanking you all for attending this morning's webinar, and we hope to see you again for some of our subsequent webinar presentations. Thank you and goodbye.